Class, good afternoon. All right. So, in our biology today, we are going to conclude on our topic, enzymes. So, this is the last lecture in the series on the topic of enzymes. Now, in this lesson, we are going to start by looking at factors that affect the rate of enzyme action. In the introduction to enzymes, we did look at some of these factors, but now I'm going to bring them here so that you can see them in their truest sense. That's the thing. Now, the rate of enzyme action simply means the speed at which an enzyme is working. So we are going to learn about factors that affect how fast or slow an enzyme works. The first factor that affects the rate of enzyme action is temperature. We already looked at temperature in the previous lessons. Now, what you must understand is this. As the temperature goes high, the action of enzymes goes faster. So the higher the temperature, the faster the speed of enzyme action. But you must be mindful because this temperature must not go beyond the optimum temperature of an enzyme. Any increase of temperature away or beyond the optimum temperature will slow down enzyme activity. We discussed this in the previous lesson. The second factor that affects the rate of enzyme action is the level of pH. Again, we discussed this matter in the other lesson where we discussed on optimum temperature and optimum pH. So, the best level at which the speed of an enzyme is at its highest is the optimum pH. So, if you go lower than the optimum pH, the enzyme action will be slow. If you go higher than the optimum pH, the enzyme action will be slow as well. So, temperature as well as the level of pH affect the rate of enzyme action. The third factor that affects the rate of enzyme action is substrate concentration. Remember, a substrate is just a substance that is being worked on by an enzyme. Now, if substrate concentration is higher, the rate of enzyme action will also be high. In other words, the speed at which an enzyme will be working when the substrate concentration is high is much faster. In contrast, when the concentration of substrate is lower, enzymes will work much slower. That's the thing. So the lower the substrate concentration, the slower the rate of enzyme action. The fourth factor that affects the rate of enzyme action is enzyme concentration. Okay? So, when you have a higher concentration of enzymes, the rate of enzyme action will be much faster. But when the enzymes are lowly concentrated, the enzyme action will be much slower. So these are some of the factors that you must understand when it comes to their effect on enzyme action. Once this has been understood, another important aspect of this topic that you must understand is the comparison between enzymes, which are biological catalysts, and other catalysts that are non-biological. In the introduction to the topic on enzymes, I did mention the fact that Enzymes are biological catalysts because they are made from living tissues. Now, non-biological catalysts are catalysts that are not made from living tissues. Now that you've understood this brief background, we can start this discussion here by stating that a catalyst is any substance that increases the speed of a chemical reaction without itself being changed in the chemical reaction. So, when you start a chemical reaction and then you use a catalyst, what a catalyst will do is that it will increase the speed of the particular chemical reaction without itself, taking part in that chemical reaction. Because it does not get affected by the chemical reaction, it remains the same as it had started. So that is the concept of any catalysts. Now, enzymes are catalysts that are made from living tissues. Because enzymes are made from living tissues, they are known as biological catalysts. Any other catalyst that is not made from living tissues is known as a non-biological catalyst. That's the thing. Now, what you must understand is this. Both enzymes and non-biological catalysts are similar in that both of them increase the speed of a chemical reaction. That is how similar they are. So when you are doing a comparison between enzymes, which are biological catalysts, and non-biological catalysts, you must know that both of them increase the speed of a chemical reaction without they themselves being changed. That's the thing. Now, since we've looked at the similarity between enzymes and other non-biological catalysts, it is now very important for us to understand the differences between enzymes, which are biological catalysts, and other non-biological catalysts. So, on your left side, we have a table that talks about the nature of enzymes, and then on your right hand side of the table we have we have a list that describes the nature of non-biological catalysts. Because we have these types of catalysts on both sides, we can easily differentiate between them. The first difference between enzymes and non-biological catalysts is this. Enzymes are proteins in nature, while non-biological catalysts are not proteins in nature. Many of them are metals. For example, iron can act as a catalyst. Manganese can act as a catalyst, and many other metals that are used in chemistry as catalysts. But enzymes are not metals. Instead, they are made up of proteins. The second difference is this. Enzymes are destroyed at high temperatures. We learned about this. When you expose an enzyme to higher temperatures, it denatures. In other words, it loses its shape. When it comes to non-biological catalysts, they work very well as you increase the temperature. So, for non-biological catalysts, the higher the temperature, the better they work. The third difference is that enzymes are made from living tissues and therefore they are organic. I just talked about this when I was talking about the comparison between enzymes and biological catalysts. When it comes to non-biological catalysts, they are made from non-living materials and therefore they are inorganic. So when you are giving this difference, you can just state to the Examination Council of Zambia that enzymes are organic while non-biological catalysts are inorganic. We know what you mean when you say the word organic, and we also know what you mean when you say the word inorganic. That's the thing. The final difference is that enzymes are specific. They can only catalyze particular substrates or class of substrates. So each enzyme is very specific to the substrates that it works on. But when it comes to non-biological catalysts, these are not specific. They can be used in many chemical reactions. So that's the difference between biological catalysts, which are enzymes, and then non-biological catalysts. So as you are learning to compare biological catalysts, which are enzymes, from non-biological catalysts, these are some of the points that you must mention. All right? Yeah. Now, as you are studying this topic, as you are trying to conclude the topic on enzymes, you must know this as well. We've got two main classes of enzymes. We've got two main classes of enzymes. There are those enzymes that work inside of cells. Those enzymes that work inside of cells are known as intracellular enzymes. Intra means inside. Cellular means a cell. So when we say intracellular enzymes, we mean enzymes that work inside of the cell. So this is the first class of enzymes. The second, class of enzymes is made up of those enzymes that work outside of the cells. So those types of enzymes are known as extracellular enzymes. Extra to mean outside, cellular to mean a cell. So when you are learning about enzymes, don't forget to understand the two main classes of enzymes that we have. Yeah. So once we are done with the comparisons between enzymes and non-biological catalysts, we can now look at how enzymes are applied in industry, how useful enzymes are in our day-to-day livelihoods. Yeah. So you are going to understand that enzymes are used in industries in the following ways. Number one, enzymes are used in baking. Without enzymes, the process of baking would have been very difficult. Let me explain how this works. When a person is baking, flour is used, and that flour is usually mixed with enzymes such as those that are found in yeast or baking powder or baking soda, if you like. Now, what happens is some of those enzymes in the yeast convert the starch, because you know that flour is a carbohydrate. Because flour is a carbohydrate, you find starch in carbohydrates. So you find that some of those enzymes that are found in the yeast convert this starch to glucose. Now, this is a very important process in baking. And then in that yeast, there are other enzymes that will now convert this glucose to produce a very important gas in baking, which is just carbon dioxide. So when this carbon dioxide has been produced, it makes that dough to rise. This dough is that, part of flour where you mix it with water and then you knead it together so that it makes, that ball of flour that we call a dough. Now, if we didn't have enzymes, this dough would not rise. And you know the implications if the dough doesn't rise. It means many of our women would make losses because their flour wouldn't raise to a point where when they sell their fritters or buns or whatever it is, that they are baking, they would not get much from it. So in action, this is what you're going to see. If you take your flour and mix it with stuff without adding enzymes to it, it won't rise. It will remain the way it is, as you can see from this picture. But when you add enzymes to it, this is what happens. As you can see, this dough of flour has risen. In other words, it has grown in size. So you find that just a little use of flour will yield a lot because of the enzymes that have been added to the flour. So you can already see one of the important applications of enzymes. The second application of enzymes in industry is found in brewing. Now, this is a big deal, okay? Before I can talk about brewing, let me explain, first of all, what this brewing is. So, brewing is the process by which beer is produced. Now, we know that a lot of our people in Zambia take a lot of beer. Now, when beer is being manufactured, enzymes are involved. Let me explain to you how these enzymes are involved. Now, when beer is being brewed, the first process is that carbohydrates such as maize or barley or wheat, any carbohydrate can be fermented. So what those people that make beer do is that they'll get these carbohydrates and ferment them. Once these carbohydrates have been fermented, enzymes are added to convert those carbohydrates into sucrose. This sucrose is a type of sugar that is very, very important in the manufacturing of alcohol or beer. Now, once this has happened, the enzymes then break down this sucrose to alcohol during the process of respiration. So you can see that without these enzymes, the most important component of any beer, which is just alcohol, cannot be produced. So without enzymes, you people that drink beer cannot have Mossy or Castle or Castle Light or Black Label or whatever it is that you drink. So you can see that enzymes play a very important function when it comes to the brewing process. The second application of enzymes in industry can be found or is found in the manufacture of washing soaps and washing powders. Now, washing detergents contain enzymes that digest and remove dirty stains from clothes. So, if you go in a shop today and then you want to buy yourself a packet of Boom detergent paste, for example, when you look at the container of your washing soap, you will find this statement written. As you can see here, they've said new, easy dissolving with multi-enzymes. So these enzymes are added to your washing powder so that when you are washing your clothes, the enzymes that are found in the washing powder will go on your clothes and then they will digest all those plant and dirty stains that make your clothes not to look good. Without enzymes, washing powders and all detergent pastes would find it very difficult to clean up whatever it is that you are cleaning up. This use of enzymes is not just limited to washing soaps and washing powders. Those of you that use detergent pastes such as Sunlight in the kitchen or whatever it is that you use, those detergent pastes are packed with enzymes. That is why you find it easier to clean your plates when you are using that particular product. So you can already see another very important, application of enzymes in industry. the final, application of enzymes in industry that we are going to discuss is this. Enzymes are used in the process that we call tanning or tanning, if you like. So this tanning is the process by which animal skins are processed so that they can be turned into leather. We all know what leather is. Now, leather is used for many things. But before we can talk about that, let me explain how this happens. So, when the people are making leather, this is what they will do. They will get the skins from animals, and then those skins are softened by using tannic acid. So this tannic acid is where the name tanning comes from. Tanning from tannic acid. Once the skins have been softened by tannic acids, enzymes are then added to those animal skins, and in this way, those animal skins are turned into leather. Now, leather is used to produce so many other products. Leather is used to produce bags, wallets, belts, shoes, and many other items that I cannot exhaust with the time that I have for this lesson. So, when you are studying this topic on enzymes, make sure you understand where we come from, understand all the aspects that I've taught you up until this far. So, this marks the end of the topic on enzymes. Where you're not clear, get your phone, call me up, or text me on WhatsApp, or reach me however way you can, so that I can explain any concept that you may not have understood. That's the point. So we've come to the end of enzymes, which is our topic number 3 in biology. In the next lesson, I'm going to teach you how you answer exam questions when it comes to enzymes. With this said, let me wish you well in your studies. Let me wish you a good day as well. I'll see you again later.